Well, we're ready to finally begin what most of you took the course for, was to examine some anatomy and physiology. We've been doing a lot of chemistry, biochemistry, tissues, cells. We're ready to embark on the first of 11 organ systems. And I'm talking about the integumentary system, which is chapter 6 in our book. So, of course, you recall from chapter 5 that we have 4 major tissue types found in our body: epithelial, connective, muscle, and nervous tissues, which, if you remember from chapter 4, is made up of cells that perform a particular function. So if you remember our first lecture, we talked about a hierarchy of organization within the body, starting with the atomic level and working our way up through organism. All right. Well, we're up to the organ level here and organ system level. And so when we talk about an organ, be that the stomach, the liver, the lungs, the heart, it's a more complex structure than a tissue is. In fact, an organ is typically made up of a variety of different kinds of tissues. We saw and have talked about how in the small intestine, for example, we have lining. The innermost lining of our small intestine is simple squamous, right? That has those microvilli to absorb more efficiently those various organic compounds that we're digesting. We also talked about how if we go a little bit deeper within the lining of the small intestine, below the simple columnar cells, we would encounter smooth muscle. And we said that smooth muscle helps to push food through that small intestine in a process referred to as peristalsis. So there's a good example of how an organ, i.e. the small intestine, is composed of an epithelial tissue as well as a variety of different kinds of connective tissues, not the least of which is smooth muscle. I'm sure we find fat there as well. And as we then go a step above organ, we can talk about a system, an organ system, of course, composed of a variety of different kinds of organs. So again, think of the digestive tract or digestive system. It's made up of not only the small intestine, but also the large intestine, the stomach, the esophagus, and so forth. So, as I said earlier, we're ready to embark on a discussion of our first organ system. But I want to ask you a question first. What's the largest organ in your body? Any idea? You might think maybe liver, if you think about the rat dissection and how large that organ was when we opened up the abdominal pelvic cavity. It's a really big organ, certainly one of the largest in the rat. But the reality is the biggest organ in your body is the skin. Yes, your skin is an organ made up of tissues, which we're going to talk about. In fact, a considerable percentage of your body weight is skin, 15%. If you were to weigh yourself, multiply that by 0.15, that would give you the pounds of skin on your body. And of course, we know what organ system that is in, our integumentary system. We talked about functions of the skin when we discussed histology. If you recall, the skin is composed of stratified squamous. So one very important function, of course, is in protection, right? Protection from pathogens, things like bacteria, viruses, certain kinds of fungi, all could potentially cause infection. And if we can prevent that pathogen from getting in, then we can thank our skin for that effective barrier that it provides us. It's not the only function. If you think back to our very first lecture, we talked about homeostasis and the use of negative feedback mechanisms to ensure a stable internal physiologic state And we gave the example of body temperature, right? The fact that receptors can pick up on increases in body temperature, for example, in turn stimulate the control center in our brain. The hypothalamus of our brain is where our little thermostat exists. And that thermostat is going to compare that stimuli coming from the thermoreceptors of our skin to 37 degrees Celsius or 98.6 Fahrenheit and realize that the thermostat has been elevated. And in response to that, what do we want to do? We want to negate the rise. So the control center in turn stimulates, of course, different effectors like sweat glands, right, which are part of the skin. And we begin to perspire or sweat. And thanks to evaporative cooling, we lower our body temperature, right, bringing it back toward a more normal range. So here's a really interesting electron micrograph found on the very first page of chapter 6. And it looks as if we're looking down into a forest from above. This, of course, is actually showing hair shafts. Those are the trees, if you will, amid the surface stratified squamous of our skin. So that's a pretty amazing photograph when you stop and think about it. This is a histologic stain section, which we'll be looking at in lab very soon, of the skin. And let's begin to define a few different areas here. At the very superficial most area there of the skin is our what? Stratified squamous, right? Yeah. Now you don't see that apical surface of the cells. It would be somewhere up above. I kind of wish they would have a little white space up here to indicate, you know, where the air, if you will, would be, that open surface that we know all epithelium must face. And we'll talk about the different reasons why we have different colors here. But that's stratified squamous. This would be where the basement membrane would be, where I'm moving my cursor, my arrow. Okay, which actually extends down. We'll talk about what that is in a few minutes and then back around. Below the stratified squamous, we have a fairly wide, thick range of structures here, the most abundant of which is dense, irregular connective tissue. We talked about that in the previous chapter and also a bit in lab. Remember, this is composed of primarily collagenous fibers, but their orientation is a bit more haphazard, if you will, or random, unlike the dense connective tissue, say of tendons or ligaments, where we have dense parallel bundles of these strong collagenous fibers. Here, they're much more loosely oriented. And if you look closely, you'll see down below here another kind of connective tissue. Hopefully you can recognize that. That, of course, is adipose tissue, another type of connective tissue. So again, if one knows what to look for in a histologic section, and this takes practice and years of experience, I guess, but if you can take the time to look and you know what to look for, you can see a variety of different kinds of tissues in a slide like this. Here is an artist's sketch of a little chunk of skin. Again, no skin will really look like that in reality, of course, but like the composite cell diagram we talked about a few weeks ago, what the artist has done is he or she has basically sketched all possible structures one could theoretically find in a small little square millimeter chunk of skin. And what we should notice right off the bat is the fact that skin is composed of two primary layers: an uppermost epidermis, which is stratified squamous. This, of course, is the free surface side. This was where we were looking down from above in that previous electron micrograph, remember? These were the trees, if you will, and this was sort of the bottom of the trees, the soil, in that photo. And then as we go deeper below the epidermis, we encounter this thicker region called the dermis. And this was where we saw the dense irregular connective tissue, if you remember. And below that, we encountered some of the fat, of course, shown here in yellow. And we talked about how there's quite a bit of fat just under our skin, right, which helps to provide insulation for us in maintaining our constant body temperature. But I do want to point out the fact that this layer that lies again below the dermis, called the subcutaneous layer, or some books will call it the hypodermis, is not considered a layer of the skin. So the skin is composed of two main layers, the uppermost epidermis and the deeper dermis. And while this can give you the impression that the delineation between the dermis and the subcutaneous layer is pretty obvious here with respect to where the fat kind of begins, as I've noted here, this is really not always the case. You can have some fat cells that are found here in the mid to deep dermis. So unlike the basement membrane, which we said was fairly easy to distinguish visibly on a stained histologic section, because we said that once we kind of get past that, the look of the tissue just changes really, really remarkably and pretty obvious and conspicuous. And I think you'll agree that it is a pretty obvious switch from purple to light pink. That is not the case down here. It's a more diffuse boundary between the dermis and the subcutaneous layer. And again, we're going to talk about a lot of these various structures found in the skin. I'll just kind of throw this listing here on the right up at you. Don't let this get you all flustered. We're going to talk about all of these, but this again would be a very nice diagram to reference if you want to come back and see sort of everything in one fell swoop. This is a really good slide to look at. Okay, we're going to start with the epidermis, and then we'll work our way down into the dermis and talk about these various structures. You already know that the epidermis is composed of stratified squamous epithelium. You've seen this with your microscope and a couple of slides. I want you to note, as I've indicated on the slide, that there are no blood vessels in the epidermis. It's devoid of arteries and veins and capillaries. You don't see anything there. And of course, we have the artist sketch and labeled structures, and this, of course, is the histologic stained, same view. And I want to point out that this whole series of cells, ranging from the light yellow all the way through the white and the darker, a little bit darker, and then on into the sort of the orange peach area, and then finally terminating in this dark brown layer. This is all epidermis. This is all stratified squamous epithelium. Okay, and the basement membrane, as I've noted here, is would be at the base of these brown colored single layer of cells. So basement membrane comes around here. It would come back up around here as well. So this material in light tan, of course, would be what? Anything below the basement membrane, we said, was connective tissue. In this histologic stained section, again, where I'm moving my arrow, my pointer, this is defining the basement membrane. Everything above it in darker purple, all of this, and even up into the light purple. This is all epidermis, all stratified squamous. So the dermis, again, lies below the epidermis and is containing a variety of different kinds of connective tissues and different structures. These little finger-like projections, you see two of them here, but I'd actually have a third one over here and the beginning of a fourth one, which is cut off. You can't see it here. These are called dermal papillae. If I'm talking about one, I would use this word, papilla. If I add an E at the end, that is a plural term. And dermal meaning it's in the dermis. A papilla is a finger-like projection. And indeed, these sort of look like little fingers that extend upward into the lowermost region of the epidermis. And these dermal papillae can contain a variety of different structures. I'll just mention a couple of them right now. They can sometimes contain blood capillaries. Now remember, I said a moment ago, the epidermis lacks blood vessels, but we're in the dermis here. So the blood vessels can make it as far north, if you will, as the dermal papillae. They can get right next to the basement membrane. And they're typically found, the blood capillaries are, in these little finger-like dermal papillae. The dermal papillae can also contain a touch receptor referred to as Meissner's corpuscle or a tactile corpuscle. And we'll talk more about that in just a few moments. But I think what we're seeing actually right here, where I'm circling with my pointer, the arrow, this is actually looking to me like a Meissner's corpuscle, which would be connected to a nerve fiber. And so when an insect, let's say, crawls on the surface of your skin, like a mosquito, you can tell that there's something crawling there. You sense that insect thanks to the displacement of the insect on your epidermis, causing this little Meissner corpuscle to be moved, to be displaced. And when that gets moved or displaced just a little bit, it triggers a nerve impulse that travels to your brain that says, I have a mosquito about ready to bite me. Or a warm breeze you can feel on your skin, right? That's because you are stimulating those very, very sensitive Meissner's corpuscles housed high in the dermis, right next to the epidermis. We're going to talk about various layers now of the epidermis. So again, not trying to confuse you, but skin made of two major layers, epidermis and dermis. And now let's talk about the sub-layers. I guess they don't use that word sub-layer, but it's a subset of the epidermis. There are 5 layers of the epidermis. And we're going to start, as I've mentioned here, at the deepest layer, okay, which is going to be this brown set of cells, colored cells, or in the case of the photograph, it would be these that I'm kind of tracing just above the basement membrane. And then we're going to work our way up toward the apical surface, okay? So this first layer, this deepest layer of the epidermis, is literally 1 cell layer thick. It's called the stratum basale layer. Some books will actually refer to it as the stratum germinativum. I'm going to stick with stratum basale layer, easier to say. And this particular single layer, the deepest layer of the epidermis, is composed of cuboidal or columnar shaped cells. Now, again, I'm not trying to confuse you because we technically define all of the epidermis as stratified squamous, but they are, we'll just call them cuboidal or columnar-like cells. And these sort of give you the impression that they are not flattened, are they? But they are a bit cube-like. What's really interesting and something you need to remember about this particular layer is that these cells right here are actively undergoing cell division, which we talked about in chapter 3, right? The parent cell undergoing cell division to give rise to the 2 daughter cells. That's happening on a 24/7, 365 basis in you and I. And those cells that are produced, they become cells of these various other layers that we're going to be talking about. In other words, these cells get pushed literally toward the surface, where they eventually die and are sloughed off. You lose thousands and thousands and thousands of skin cells every day, and those you're losing here are at the very, very top of the epidermis, but they have as their origin this layer, the stratum basale layer, the deepest layer of the epidermis. That's where they were born, if you will. Superior to, or superficial, that's a better word, superficial to the stratum basale layer is the thickest layer of the epidermis called the stratum spinosum. Many, many layers of cells located here with large oval nuclei. there are also fibers of keratin beginning to form in this layer. And what the protein keratin acts to do is to provide a waterproofing characteristic to our skin. I also often refer to it also as sort of the Vaseline intensive care lotion of the skin. It keeps the skin rather pliable and moisturized, if you will. But keratin is a very common protein that is manufactured here in the stratum spinosum layer. And as these cells undergo this keratinization process, the acquisition of the keratin, they're eventually going to harden and eventually die. Toward the upper layer of the stratum spinosum layer here, notice the cells are beginning to take on a more characteristic squamous shape, aren't they? Yeah. Above the stratum spinosum is the stratum granulosum. This is 3 to 5 layers of flattened granular cells that have begun to shrink. They have keratin inside them. Their nuclei are also beginning to shrink a little bit. So that's that keratinization process that I was referring to a few moments ago. The keratin actually gets embedded within the cells. Stratum lucidum is a single layer of rather clear cells. In fact, it's really tough to see the stratum lucidum layer in a stained histologic section, and they have it, denoted here above the dark purple, which would be the stratum granulosum layer. So it's really, really hard to see them. They're very, very clear, and they're very, very flat. And there's just one layer there in the stratum lucidum. And then finally, the superficial most layer of the epidermis, that which actually has a free surface to it, is the stratum corneum, shown here in yellow. Here's that white space, that would be the air around us. And unfortunately, they don't show that white area here in the histologic section, but it would be right above that. This is that characteristic stratified squamous that we would have recognized probably in the slide we looked at last week in lab. But by the time the cells get to this region, this layer, they're fully keratinized and they're dead. So these cells are dead. And these are the ones that get sloughed off all the time. But remember, they're getting replaced thanks to the cell division going on in the stratum basale layer. What's really fascinating to me is how thin the epidermis is. And you have to kind of understand that it depends upon where one goes on the body as to how thick or thin that could be. If you look at the soles of your feet or the palms of your hands, where we tend to have more wear and tear due to use, we might have a little bit thicker layer of epidermis, but if you look on the back of your hand or on your face, that epidermis averages, as it mentions here, about 0.1 millimeters in thickness, if I just use that as an average. Now, you've got to remember the metric ruler that you used, the second lab where we did the prediction of height based on femur length, remember that? We measured the height and we measured our ulna length in centimeters using the meter stick, but I also had you pull out those small rulers. There are 10 millimeters, remember, in a centimeter. So a centimeter is only about yay big. It's really small. And we got 10 subunits, those are millimeters. I'm suggesting that we take 1/10 of one of those millimeters to define the average thickness of the epidermis. So it is amazingly thin. We also just finished talking about the active cell division going on here in the stratum basale layer. And that process is fairly constant over time, so that the cells that you lose every day are being basically replaced thanks to that cell division going on in the stratum basale layer. I want to point out another interesting cell that we find in the stratum basale layer of the epidermis. And let me again sort of reorient you here a little bit. We're not looking at those 5 sublayers of the epidermis, although I know here at the top, of course, would be which layer? There's the apical surface, so this is stratum corneum, right? Then I'd have stratum lucidum, and I'd have stratum granulosum, and I'd have stratum spinosum, and eventually here at the deepest layer of the epidermis, right next to the basement membrane, what is this layer of cells called, remember? That undergoes active cell division? Stratum basale, right. So just understand we have all those 5 layers shown here where it says epidermis. In the stratum basale layer, we will occasionally find a special kind of cell noted here, which looks sort of like an upside down octopus. This is a melanocyte. And this melanocyte has an interesting shape to it, doesn't it? You can see, of course, the organelles. Here's some mitochondria, Golgi bodies, big, weird-looking nucleus. It also has cytoplasmic extensions that resemble tentacles that extend upward, don't they, into the stratum spinosum layer. And inside those little tentacles, quote-unquote tentacles, those little cytoplasmic extensions, are little brown pigment molecules called melanin. There's our melanin pigments. And these cells, these melanocytes, can actually deposit that melanin in some of these cells of the stratum spinosum. As you can see here, there's like 1, 2, 3, 4, 5, 6, 7 cells here that have deposited melanin that came from this melanocyte. Not only that, but the melanocyte itself, of course, has melanin in its little cytoplasmic extensions. We're going to talk more about melanin in just a few minutes, but that's an important pigment. Okay, let's talk about what does melanin do. Here's an electron micrograph showing a single melanocyte. That's that kind of funny wavy looking nucleus, all right? We don't see any of those cytoplasmic extensions, the little octopus legs or anything like that. But we can see some of the pigment granules here that have been stained black. That would be the melanin pigment itself. So what's the purpose of having the melanin? I think I refer to this as a scanning electron micrograph. It's not. It's a compound light microscope, really magnified a lot. What does that melanin do? Anybody know? Well, it protects us. It also gives us our own unique skin tone or skin color. Yeah. Now, one's skin color is dependent upon how much melanin those melanocytes produce. Somebody that has very dark complected skin, someone of, let's say, African American descent, would have a lot of melanin in their skin because the melanocytes that are there are producing copious amounts of the pigment. In somebody who is of, let's say, Caucasian ancestry or Caucasian race, of European ancestry, I guess, is probably what most of us can trace our family trees back to some part of Europe. We're more light complected because our melanocytes don't produce as much melanin. But that amount of melanin that our melanocytes do produce is determined by our genes. And sometimes you run into some interesting genetic disorders whereby the individual does not inherit the proper genes from his or her parents that regulate melanin production. And so here's a child who lacks any melanin at all in their skin. Their skin is very, very white. And their iris, the colored part of the iris here, is extremely light, very lightly colored, a light blue or very, very, very light green. And that's due to the lack of melanin. So it's technically a mutated gene. And somebody with albinism, it's called, has to be very careful. Even going outside on a sunny day really increases their risk of getting burnt or hurt by the sun. The sun's UV light can be extremely harmful to somebody who is suffering from albinism. So some of these folks only go out after the sun has set, or they spend their entire social lives out when it's dark, because they literally cannot go out during the time the sun is out. Or if they do, they've got to really cover their bodies. It's in real severe albinism, that's the case. It's kind of crazy. So in addition to giving us our own unique skin color, skin tone, based upon, again, the amount of melanin produced by our melanocytes, there's another thing that our melanocytes do, and that's to protect us from UV rays. But how many people use tanning salons to get that nice, dark, deep tan, right? You're getting ready to go on a vacation, and you want to get a tan so you don't burn when you get to Barbados or wherever you're traveling. Or you spend time out in the sun in the spring, right? Because our society sort of has embraced having that nice, dark, deep tan. It's a very sexy look, right? Youthful look. You look good with a tan. But we do that at a risk to ourselves. Because if we expose ourselves to too much UV over time, or even a short, bad sunburn can be problematic. Okay, it doesn't have to be necessarily, you know, through your teen years or your 20s and to your 30s that eventually this sort of catches up to you. Even intense sunburns can really lay the groundwork for potential skin cancer. We'll get into that in just a few moments. But you've seen people who have tanned since they were kids, and now they're in their 30s and their 40s, and they kind of look like this. You know, their skin is really looking bad. Looks like cowhide. This obviously is the extreme, but you need to be conscious of this risk you take when you are tanning, where you're exposing yourself without using adequate sunscreen. Because the UV rays from the sun is what causes our melanocytes to produce more melanin. That's why you tan. When you subject the skin to UV, be it the sun or tanning lamps, it causes the skin to produce melanin. The melanin is there to protect you from the UV rays effect. But, as I said, you risk getting skin cancer if you subject the skin to too much UV. And this is one particular type of skin cancer. They're collectively called the cutaneous carcinomas. And there's a clinical application box you should be checking out there in your book that talks about skin cancer, and then these two in particular. These are the two most common types of skin cancers, the basal cell and squamous cell carcinomas. These carcinomas arise from non-pigmented epithelial cells. Okay, so these would be cells in the mid to deep epidermis that begin to mutate as a result of having been bombarded with UV. The UV rays of the sun can induce genetic changes, and those mutations that we talked about in one of the earlier chapters, when that cell goes to divide to make the daughter cells, those daughter cells will carry that mutation, and so on and so forth. So you can eventually have these growths forming. These, and they're kind of like tumors. The upshot, if you want to talk about the silver lining, I guess, of these two, which, you know, they like to be fun to have. The only advantage of having these two types of skin cancers versus the one we'll talk about in the next slide is the fact that they're typically slow growing, as I indicate here, and there's a fairly high cure rate if you catch these early enough. Now, if you have a basal cell carcinoma on your back and you never see your back, and it sits there for months or years, it can develop into a much more serious life-threatening type of skin cancer. But assuming that it's seen, it's picked up during a physical, or somebody notices it, or you see it on yourself, and you go into the dermatologist and you have it biopsied, where they take a little sample and they can confirm whether it's basal or squamous cell carcinoma, then there again are a number of surgeries that one can have, different kinds of inpatient surgeries where they'll remove the affected area and probably down a little bit deeper to make sure they've got the whole cancer. Or sometimes just irradiating it can also destroy those particular mutated cancerous cells. So these are the common ones. These are the ones that people get, that farmers get, or anybody who's outside a lot. Somebody who works construction is subjected to sun for a large amount of their working life. And this is why we always stress the need to wear sunscreen when you go out. Or even in the middle of the winter, when there's snow on the ground, you get that double reflection when you're out skiing or snowboarding or snowmobiling or whatever. You should really think about that. You can get just as bad a burn in the winter as you can in the summer under certain conditions. Summer, of course, being more common, but you should always be wearing sunscreen. The second or third type of cancer is the one that we need to be most concerned about, called malignant melanoma. Now, notice that it doesn't make up does not make up a great percentage of skin cancers. Less than 5% on average are of this type, but notice the high mortality rate. 80% of skin cancer deaths are from malignant melanoma. Now, if you look at the word melanoma, what does that remind you of? We talked about a word not too long ago, a particular cell that produces melanin, the melanocyte. Remember the melanocyte housed in the stratum basale layer of the epidermis. Those are the ones that have the tentacles, if you will, the cytoplasmic extensions that came up. It's here in these cells that the mutation occurs as a result of that sun exposure. So these are the cells that are dividing uncontrollably. And that's one reason why they look darker in color, because of the melanin that's being produced in copious amounts at those particular lesion sites. Malignant means, remember, that it can spread, it can metastasize. And that's the problem with malignant melanomas. They can spread throughout the body and set up shop, if you will, in other organs and lead to bad outcomes. These are the types of skin cancers that are often caused by intermittent exposure to high intensity sun. And so again, I'll get on my high horse and say, make sure when you're outside in the summer or even in the winter under certain conditions that you consider wearing sunscreen. I'm not talking about sunscreen number 2, I'm talking about sunscreen number 30 or 40 to really prevent the penetration of those harmful UV rays from the sun. Because you do not want to get this. So how does one recognize malignant melanoma? Well, dermatologists have come up with a fairly easy way to visually differentiate this very nasty skin cancer with more benign moles. We all have moles. Those are not cancerous. So a malignant melanoma, unlike a mole, is going to be asymmetrical. What does that mean? Asymmetry. The prefix a means without. Without symmetry. What is symmetry? If you take a circle and you cut it right down the middle, you have two identical halves that are symmetrical, right? Take a square, fold it in half. You have two symmetrical rectangles. So mirror images, if you will, of one another. That's kind of what symmetry means. Cut you down the middle, right? Mid-sagittal section, right and left equal sides. If you look at that preceding set of photographs, you'll see that those malignant melanomas, those dark pigmented lesions, are not symmetrical generally. They're asymmetrical. They have a very different half if you tried to slice them, virtually down the middle. If you look at the border of a malignant melanoma, you'll find that it has often scalloped or irregular borders. A mole will have an even border. So B, another characteristic one could look at in addition to asymmetry, look at the border. If it's scalloped or has irregular edges, that can be a warning sign. Actually, it can be a warning sign not only of malignant melanoma, but even some of these other ones. The cutaneous carcinomas also, have some of these properties as well, although we're talking about malignant melanoma specifically right now. Color. Well, as you know, the malignant melanoma typically is very dark, pigmented, brown to purple to black. Moles typically don't have that particular variation in different colors. Usually one shade versus multi-dark shaded of the tumor. Diameter, the size. Most moles are smaller than 6 millimeters. And again, if you want to get your ruler out, you can see what 6 millimeters is. It's a little over half a centimeter. Some of these melanomas on the skin can be very, very large, like centimeters in size. So just overall size is a pretty good indicator too. And the last one, E, evolution. Here we're talking about, is there a change that takes place in the growth? Moles don't generally change in size or shape, quickly, generally. Does the color change? If the color changes or the size changes or the symmetry changes or the border becomes more irregular, these are all things that should raise the warning flag. Itching, tenderness, something that just does not quite feel right. When you scratch it, it bleeds easily. All in indicators potentially of a type of skin cancer like malignant melanoma. So as I've said at the bottom here, you know your body better than anybody else. And you really should pay attention. And if you see something that doesn't look right, don't dismiss it. Doesn't even have to be on you. It could be your mom or dad or grandfather or your aunt or whatever. Okay, we'll now move down into the deeper dermis. We've already talked about the dermal papillae, those finger-like projections, remember, that we said could house perhaps Meissner's corpuscles, those very sensitive sensory receptors, or some dermal papillae could house blood capillaries. You'll be seeing those in lab. We also talked earlier about the dense irregular connective tissue that is also found in the dermis. We can add to that areolar or loose connective as well. You saw that recently in lab. The haphazard arrangement of both elastic and collagenous fibers, remember, in the matrix. We just talked about how the epidermis was on average about 1/10 of a millimeter thick. Well, now we're talking about a much thicker dermis. However, it's relative, is 2 millimeters thick. Well, again, get your ruler out, look at that. It's not very thick at all. It's thicker than the epidermis, but still we're talking, skin, epidermis plus dermis being, 1 to 2 millimeters really in length, overall length, or thickness, I should say. So you're not too far from the surface, right, of potential pathogens being out here. If you were to cut through that epidermis and dermis, you could introduce, and sometimes we do introduce pathogens. If we cut ourselves by accident or get a paper cut or cut ourselves while we're cutting vegetables for dinner or whatever it might be, if we bleed, we've cut into the dermis. Well, I've talked about one of the two types of touch receptors or pressure receptors, and that, of course, was the tactile or Meissner's corpuscles, housed again in the dermal papillae. Okay, this is right next to the stratum basale layer of the epidermis. So because of their position high in the epidermis, they're high in the dermis, excuse me, they're very, very susceptible to light touch or light pressure. The other type here, the lamellated or Pacinian corpuscles, are located mid to deep dermis. And therefore, it would take a more significant amount of pressure on those to cause them to be stimulated and in turn send an impulse down a nerve fiber up to the brain, and we would detect that particular sensation. Also in the dermis are thermoreceptors that are sensitive to temperature changes. Sometimes just being by a hot campfire, you feel that heat, right? Thanks to hot receptors in the dermis. Likewise, you can stick your hand into a freezer and you can feel the cold. You have cold receptors that are stimulated due to that particular temperature extreme. So back to the diagram we saw a little bit earlier, here we can see several dermal papillae that are housing blood capillaries, right? Here and here and here. Here's one, here's one, here's one. Now, they don't do a super great job showing the Meissner's corpuscles, although this one is sort of situated very close to a dermal papilla. Some of these other ones are definitely a little bit deeper, and that's because they're not Meissner's, they're Pacinian. But you know, I don't like the way that they're sketching these. I'd much rather see a couple of these like way high up in the dermal papilla. So pretend this guy is way up in here, pretend there's another one right here. And those that you find sort of mid to deep, these are going to be that second type, the lamellated or Pacinian, that would be stimulated upon deeper displacement of the epidermis and upper dermis. Okay, some additional accessory structures found in the dermis. I think you've heard of most of these before, so these shouldn't be totally new, some of them anyway. Nails, our fingernails and toenails. What are they? Well, when you look at a fingernail or a toenail, you find, of course, this large thing called a nail plate. Now, this is actually composed of dead cells. Yeah, who would have ever thought? If you look at the base of the nail plate, we have something called a lunula, lunar moon. So this is like a half moon structure. At the base of all of our fingernails and toenails, we have this darker, whiter lunula. And as I've indicated here with the slide, there's active cell division taking place here in the lunula, similar to the way there's active cell division in the stratum basale layer of the epidermis. And as these cells get further and further from the blood supply, which would be found at the base of the lunula, they die, they begin to acquire these keratin fibers, and eventually become part of the nail plate. Now, you know that every few weeks you need to trim your nails. That's due to the active cell division and then the ultimate keratinization and death of the cells as they get pushed away from the lunula toward the free edge. And so what this nail plate is basically doing is it's protecting the cells here in the nail bed, it's called. Evolutionarily, and I don't want to get into a big discussion of this, but it's very likely we have nails because at one time in our previous hominid evolution, and even before that probably, we probably used our nails to dig at the soil or whatever it might be to get to tubers or whatever, right? I mean, you think of your dog and your cat, they have nails. Deer and cows have hooves, and they grow. A cow or a horse needs to have their nails clipped, trimmed every once in a while. Your dog, you know, you really should be trimming those. It's not always easy to do that unless you start them as a puppy and they get used to the Dremel or the actual snippers. Or an animal that's a wild animal will often just wear that free edge due to wear and tear. It'll just kind of wear off as it grows. But since we don't use our nails, you know, functionally, we have to manually trim them off. Otherwise, we would have very bizarre looking fingernails. Okay, let's take a look again at the skin and note that we have Hair shafts, of course, coming out of pores in our stratum corneum layer of the epidermis. And we can trace that hair shaft down in toward the mid to deep dermis, as we're doing so here. And at the very, very base of the hair shaft is an area called the hair bulb, where there's active cell division taking place. And as those cells divide and get pushed further and further away from the hair bulb, and note that there's a blood capillary here very near a hair papilla, so these cells are getting adequate nutrients to permit cell division to take place, thanks to the diffusion coming off of the blood capillaries of nutrients. As these cells die, they undergo keratinization. They've also acquired melanin from the melanocytes, and that's what gives hair its unique color. And you might say, well, I thought melanocytes were in the stratum basale layer of the epidermis. And the answer is they are. Go back up here. Here you can see the stratum basale of cells here in the dark purple. Well, look what happens. They can extend downward, can't they? And they make up what's called the hair follicle. Okay? So what I'm suggesting to you is that the hair follicle, which houses the hair shaft and hair root, this is a sheath, a tube-like sheath that is a downward extension of the epidermis. Even though this is dermis layer, this structure, this hair follicle and the hair shaft that's inside of it, and hair root, is a downward extension of the epidermis into the dermis. So just like we have active cell division, up here, we have active cell division down here, actually down here. And in the same way that we have melanocytes present that produce melanin, we also have melanocytes here that produce melanin to give these cells that become ultimately the hair root and ultimately the hair shaft, is what gives them their color. Now, as these cells get pushed further from the papilla, where there's active cell division, again, they undergo keratinization and they die. So when you look at a hair shaft coming out of your skin, this is made up of dead, epidermally derived cells. And at any given time, believe it or not, studies have shown that hair undergoes a periodic growth phase, and then it sort of rests. And then it grows some more, and then it rests. It's really quite interesting. They talk a little bit about that in your book, so check it out. Here's a stained histologic section of the part of the dermis, and to orient you a little bit here, here's the hair follicle extending down from the epidermis. This tube-like sheath that wraps around, comes back up the other side. Here's the hair root in the very center. Here's the hair bulb where there's active cell division. You don't see the hair papilla, but it would be right about here. This again talks a little bit about that growing, resting process that a given hair undergoes throughout time. This looks to me like another hair follicle, just given the similar stained nature and histologic look of the tissue. This is probably another papilla right next door. But this is a pretty interesting photograph. Take a look at it. What do we have here? Well, it's labeled, doesn't it? This is, of course, fat. This is a really interesting electron micrograph, magnified almost 900 times, that shows a hair shaft as it's working its way toward the surface of the body. These are those dead, keratinized epithelial cells that I was referring to a few moments ago that had their origin way, way deep in the hair follicle, the base of the hair bulb. And as they work their way up to the surface again, they die. It's like a bunch of dinner plates that have been stacked one on top of the other, one on top of the other, on top of the other as they work their way toward the surface. It's kind of interesting. This is probably some fat, looks like, in white. This structure right here that's attached to the hair follicle is called an erector pili muscle. Now, we just finished up talking about muscles not too long ago, skeletal, smooth, and cardiac. This obviously, as it's labeled, is smooth, meaning you have involuntary control over it. So you might say, what's the purpose of the erector pili muscle? Well, when you get cold or you're suddenly frightened, this smooth muscle can contract, it can pull on the follicle, and in turn cause the hair to stand on end. If you've ever seen those, the stereotypical Halloween cat, the arched back and the hair standing straight up, that's sort of what happens when the erector pili muscles all contract at once and the hair goes straight up. Or when you get goosebumps, goose pimples, that's due to the erector pili muscles contracting, pulling on the hair, causing it to go straight. Here's some additional accessory structures of the dermis. These, of course, are called sebaceous glands. We see that this particular one has been sort of sectioned, so we can kind of peer inside of it. But what I want you to notice about the sebaceous glands is that they are connected to the hair follicle and will release a fatty substance called sebum. Sebum. And when this sebum moves toward the surface and eventually coats the surface of the skin, it, like the keratin, provides a waterproof characteristic to the skin. I think I said earlier when I was describing, I forget what substance, and I used the term in Vaseline intensive care lotion. This is the Vaseline intensive care lotion that I was referring to, the sebum. Yeah, these oily secretions do keep the skin waterproof, but also provides sort of a waterproofing, lubricating conditioner, really, to the skin under normal conditions. Now, sometimes the sebaceous glands can overproduce sebum, and we can have issues, which we'll talk about in just a moment. But you're going to be seeing sebaceous glands when we examine some sectional views of the skin, and you'll see that they're often closely allied to, they're often connected directly here to a hair follicle. You don't always see that. It depends on where the knife cuts the skin, but sometimes you see that directly connected to a follicle. So here's another photograph of a skin section just to orient you again. Here we can see the epidermis right here. Here's the beginning of the dermis. Here's a hair shaft in yellow. Here's a hair follicle. This darker purple structure is showing a hair follicle. Here's another hair shaft in the center. But they're pointing to a couple sebaceous glands. And oftentimes these glands have cells that are quite large. They're very transparent. They don't take on the stain too much, although their nuclei are also very easy to see. So I look for large, clear cells that are clumped together with a dark, obvious nucleus. That tells me I have a sebaceous gland. So I have one here. I have a little one actually right here as well. Here's one here. You'll be able to see those pretty easily once we start looking at some slides. Here's a magnified view of a sebaceous gland. And again, look for clear cells with dark stained nuclei clustered together. Another one up here. That screams sebaceous gland. This is also situated very close to a hair follicle. That's what we're looking at here in the dark purple. And in the center of that would be a piece of hair. Your book talks a little bit about hair loss and pattern baldness there in clinical application 6.2. So I'd like you to definitely read over that short little box. I think you'll find it very interesting. What are the treatments? What are the causes of hair loss? Check it out. Also, I want you to read over the clinical application box 6.3 that talks about acne. Acne is due to overproduction of sebum by the sebaceous glands and is often triggered by puberty, because at puberty we have a sudden rise, usually in a group of male sex hormones called the androgens. Now, testosterone is an androgen, and girls produce small amounts of androgens. Guys produce more. But I want you to note here that the source of these male sex hormones, these androgens, do not come from the testes exclusively. They also come from the adrenal glands. And that's why both women and men can sometimes have acne issues, often starting at puberty, due to the adrenal glands producing these male sex hormones. And what, again, they do is they cause the sebaceous glands to overproduce the sebum. And producing a lot of sebum can oftentimes plug the passageways of the sebum toward the surface, and bacteria can begin to metabolize the fat in the sebum. Sebum is made of fatty materials. And so the whiteheads that you see here is due to the pus, not to get gross about it, but it's what it is. It's due to the pus that forms as a result of bacterial metabolism of the sebum in the overactive sebaceous glands. So what can you do if you have really bad acne? Well, you can try to use topical ointments to kind of keep the pores open so that there isn't the opportunity for the passageways to get plugged and the anaerobic conditions to basically develop, which encourages the bacteria to utilize the sebum. So keeping the pores open can be helpful. Sometimes in really bad acne, the dermatologist may prescribe an antibiotic, a systemic antibiotic, to kill the bacteria as well. It really depends on how severe the acne is. There's even surgical treatments and UV light treatments that can be used to treat acne. Most of the time, it's impacting earlier parts of one's life than mid to late age. Another interesting accessory organ, in addition to the sebaceous gland and the erectopili muscle and so forth, is the sweat glands. And we have actually two different kinds of sweat glands. I'm going to just mention the one here, and then we'll talk about the second one in just a moment. This is called the eccrine sweat gland, the most numerous of the two. And this is the one that we make use of in helping to provide ourselves with a constant body temperature. If we get too hot, this sweat gland kicks into overdrive, we perspire, and thanks to evaporative cooling, our body temperature hopefully falls, right? So this is a coiled structure that has a duct that leads toward the surface, as you can see here, exiting at a pore. So when you get too hot and you begin to sweat, you can thank these eccrine sweat glands. The second type of sweat gland is shown here, called the apocrine sweat gland. Now notice that anatomically, it looks somewhat similar to the eccrine in terms of its coiled appearance here at the base of the gland. And it too has a duct that leads to the surface. But notice that unlike the eccrine sweat gland that had a separate exit point, that the apocrine duct here eventually merges with the hair follicle, and then its secretions will be released to the surface where a hair shaft exits. Okay, so it's very different from that standpoint. It's also very different from the standpoint of the fact that the secretions from the apocrine sweat gland are not produced as a result of being too hot, where you're trying to cool yourself down. These are often being produced, these secretions, in response to a stressor situation. Like you're going in for an interview for that big job that you want to get, and you have sweaty palms, or you begin to perspire. That sweat that you're producing is not due to being too hot. It's being too nervous, really, that results in that production. So we can blame bacteria for body odor. This is what causes body odor. The production of apocrine sweat gland secretions. And here's another histologic section again that shows a couple different things. We can actually see a couple interesting structures. Here we've got an apocrine. Well, hard to say here. This might be eccrine because it has a duct that's leading to the surface. I don't see any other hair follicle. So I think this is probably eccrine sweat gland. Here's another sweat gland down below. We don't see where the duct leads to the surface. We know that it certainly does, but the knife just isn't catching it. But if you look closely, this is that coiled sectional structure at the base of the sweat gland. What do we have here? Well, we've got a hair shaft. In fact, let's take a little quick mini test and see what we can find. We've already identified the hair shaft. Right? What would be on the very surface, this sheath that wraps around the hair shaft? Remember? That's the hair follicle. What do we have here with large clear cells with distinct nuclei, often associated, always actually associated with the hair follicle, which is right here. I can see a hair follicle. Here's the hair shaft. What would these be? What would this be? This is also one of those. That's sebaceous gland. Yeah, so we can actually see a couple of really interesting structures here. Okay, so these review again the major skin glands that we've been talking about, the sebaceous glands, the eccrine and apocrine sweat glands. There are two others I want to just briefly mention. The ceruminous being one of those, ceruminous glands produce earwax. So if you ever use a cotton swab to clean out your ears, which you technically shouldn't do, doctors will tell you don't do that. But if you've ever done that, that yellow orange material at the tip of the cotton swab, that's earwax produced by ceruminous glands, which are technically modified sweat glands, interesting, in the external ear canal. And the other skin gland, which is even more bizarre, I don't know if I would define it as one, but histologically and anatomically they are, the mammary glands of the breast are technically viewed as modified sweat glands, which of course produce milk. So who would ever think that the mammary glands are technically a type of sweat gland, but they are. They don't produce sweat, they produce milk. Okay, I think I'm going to stop right here, and we'll pick up the last half of chapter 6 with the next module.